How bizarre. The current goes from screaming across the summit to 0 right now.
It's still something there. Oh, it's southwest there.
Wow, there is a really steep drop off over to the right.
Just like kind of like a
yeah, real narrow ridge we're traveling along.
I'm going to rotate slightly port, pilot.
And
heading towards those branches now so that our shore-based team, as well as Patty and I, can get a closer look at them.
I really do love the critters. Especially the fact that they decide to sit on rocks.
Yeah.
They have good taste.
And they taste good. No.
Do they?
No.
Do you eat coral?
No. No, I don't eat
Do you
coral.
eat sponges?
No. I have a colleague who tried it once.
Really?
They're fairly nasty.
That's a crazy person. Now I'm going to avoid sponge people, other than you, of course.
And Chris.
And Chris. Oh, this is a lovely spot. Look. Now, is this guy alive?
You know what?
Is this guy... Oh,
this looks like a bigger...
I've never tried to eat one, though.
This looks like what we had attached to one of the rock samples yesterday, only much bigger. Any ideas, Diva, Scott, Chris, on what this is?
To note, looks like the temperature rose maybe a degree just now. It's the first time I've seen that. This dive.
So what are the little hairy things growing on this dead coral arm?
I think there's a live colony to the left of this dead one.
Oh, you think there's a live colony?
To the left.
On the left side?
Left.
Oh, pilot.
So the things, those little things
could we look over a little bit to the left? On this particular individual, yeah. Thank you. I don't see anything, but...
Well, there's something that's whiter, but...
Okay.
They're referring to the yellow on the far left. Yeah, no, I think the dot.
Okay, so this is...
There's a yellow colony to the left.
okay.
Oh,
yellow,
the other
yellow
colony.
one to the left.
Oh, that absolutely looks alive.
So this one that we were just zooming in on is a skeleton of a dead coral, and it had hydroids attached to it.
And if you can get just a little closer.
Oh, yeah, it's attached behind the rocks with the pink encrusting corals.
Right next to it. Yep.
Yeah, it might be kind of hard. Do you want to maybe prefer to take a look at...
Would it be easier?
Looks like the same species that's up
Pilot,
at the
would
top.
it be easier for you to go to that one that's up ahead? Yeah, that's okay.
It's shy. Not like us.
Oh, and I see an urchin over there on the upper right. Beautiful. Oh, and some brittle stars there.
Oh, that's a beautiful brittle
really
star.
Beautiful. Yeah, very pretty.
I've never seen a stripy one like that.
Yeah, I've never seen one like that either, yeah.
Certainly is.
Okay, Chris, Scott, anything? Diva. Okay, so Chris is saying that this is of course, a scleractinian coral, so stony coral, and Nalipsamia. We saw some of these yesterday as well. This beautiful imagery. You can see the polyps extended.
Yeah, 7-second delay, but
Yep, I understand.
yes.
Okay, video, do you have what you need?
It seems like I'm interrupting because I don't hear you talking
Yes,
for
video
like
just needs a
7
wide
seconds,
shot.
and I try to make a comment. But Enophthalmia has got polyps typically on one side, and you can sort of see that in this colony, even though it's kind of a bushy colony.
Okay.
That's great, Pilot. Thank you.
I need to pull out wide.
And the rocks that these corals are nestled among looks different from the area we were before. These look very blocky. Sometimes the composition
there's
of
a fish.
Oh, it's
What
a
is that?
fish. I'll shut up. I'll shut up. Let's look at the fish. Woo, look at the tail. Oh, gorgeous. That is a beauty. What is that? Bruce Mundy. A little fish?
This is great.
It has a follower.
Fish. This is an onaga.
Oh, wow. Bottom fish. First bottom fish.
Oh, just ate something.
Really? Cool. I missed it. I was going to try writing
Oh,
down
isn't that
a
nice?
note.
Oh, that's really nice.
Thank you
Oh my
for
gosh,
the
that's really
beauty view.
nice.
Oh, cool.
Pull wide.
This one's
this is
very accommodating.
beautiful.
Sometimes the fish are attracted, to the ROVs, to the lights. They are curious. And fish are pretty smart.
Rotate left and get him in the dark.
Is there a common name for this?
Rotate slightly just to keep you in view, Pilot.
Pink
Pretty
beauty.
big. Yeah, pink beauty.
Of all the bottom fish that we were hoping to see, this was the primo one.
Oh,
This is
Fantastic.
How big is that thing?
There's
wonderful.
That sure was a
Oh, that's
pretty fish.
wonderful.
a the rock.
It's a nice size. Yeah, you're drifting back and down, Pilot.
Look at that. Long
I certainly
tail
am.
fin. Oh my gosh.
That was beautiful.
Excellent work, pilot.
Our pilots
Yeah,
are
that was
very
fantastic.
good at convincing fish to do their most expressive. Shucks, I'm lost for words.
Yeah. It's like temperature,
That was a
spiked
gorgeous
another
fish.
half degree when you went out the side too. A lot going on.
Here's some hefty thermoclines, I guess.
I'm going to rotate slightly back to starboard.
So that fish is called onaga.
Onaga is,
onaga, and its common name is the long-tailed red snapper. Well, and you were asking how they get names.
Yeah.
You called it pink beauty.
A pink beauty, yes, but in Hawaii, those are some of the prized fish for eating. So we will let this one go along its merry way. Have a nice life, fish. Have a lot of babies, please.
So it's trying to push you into the cliff right now.
Shirley, now there's another one of those round sponges.
Yeah, but it's the same. Very
Same guy?
yeah, really similar.
not
It might be a little bit different, but it's not. It's fairly similar.
Okay.
15.
Fine. Yes.
Okay.
Fine.
Yeah.
Where are we? 366.
Maybe 220. 15.
15 at 220. Okay.
You can do maybe even 20 if you want.
So let me just
20
explain
Yeah.
for folks at home as
Yeah,
we're
so
moving
20
along.
to 20.
20,
The setup
20,
for the
0.2.
vehicles
All twos?
is
All twos?
twofold. We have the ship, of course, on the surface with a tether that permits the pilots to send signals down to the
Bridge
remotely
already has.
operated vehicle, which is the one that has the camera on it that you're seeing the video from. But between the two,
Hey, we're gonna get a
there
ship
is
move in.
another vehicle that is called
All right,
Sirius.
we
So
want 2
if you
meters at
hear us
222
mentioning that term,
degrees at 0.2 knots.
that's the vehicle that is above it. Well, now we're going to zoom in on.
Copy that.
So another twofer. So we have an
Yeah,
urchin,
an
and we
urchin,
have
right.
a sponge.
That's another sponge.
That sponge is probably Geodia. Beautiful.
So what is the urchin doing on the sponge? It's not eating the spicules.
Well, no, it's not eating the spicules, but there are some echinoderms, and urchin is part of that phylum, that actually do eat sponges. But so this could be eating, grazing what's on the surface of this sponge. But as you can tell, there's not a whole heck of a lot on the surface. But it could be grazing on what's on the surface of the sponge. But it could also be taking some little nibbles out of the sponge.
Ah.
So Mary Jo Brown says that she's seeing fractures through the boulder that these sponges, the sponge and the urchin are on, and the fractures, she says, look radial. So this might happen as the pillow lava cools and contracts.
That's absolutely right.
And this is a different urchin, type of pencil urchin. You look at it really closely. You can see its little tube feet.
Those are the thin ones that are moving back and forth.
Yeah, down on the
Right.
bottom. Yep, and
Right.
they can use, the urchins use that to kind of move along.
Plenty for them to eat down here. So as we zoom out, I am going to take a look at those fractures. Oh, yes.
Partial.
You can see fractures that they look actually as if they're radiating from the point where the sponge is. And that's very, very typical of cooling cracks in pillow basalt.
So we're seeing some more close-ups of this stoloniferous octocoral that we saw very abundant earlier on. Slightly deeper, we're seeing tubes, probably polychaete tubes, those white
Hair-like things.
Yep.
Yep. They're everywhere on the rocks. So this may be a little mound of lava that squirted out along this ridge. And as it hits the seawater, it will cool quickly to form a glassy crust.
So Asako Matsumoto is joining us from Japan, and she gave us, because
Side and back
Patty, you were
and
asking about the names of these organisms, and what she tells us is that in
Left of lasers.
Japanese,
See
the,
that
for the onaga,
Yeah.
O means tail, and naga means long, so long tail.
Oh,
So
how
the Hawaiian
perfect.
common name of onaga is long tail.
All right.
We still got about
Thank
2
you,
meters left
Asako. That
or
was
so,
really
Joe.
nice of you to call in.
Yeah, I have you about 70 in my camp.
She's logging in on our event log.
Yeah.
So at the top
you
of
can
this
look.
mound, we're
No
seeing
problem.
some more Geodia sponges.
Although we might
And
have a little
lots
bit of more
and
tether
oh, wait a minute.
for
Isn't
current
that a
to take.
crinoid?
Copy that.
I don't
think-
see what you're.
I'm looking at that thing.
Oh yeah. There.
Did I get it?
You did.
Hey, I called it. It just proves that old dogs can learn new tricks. Now, what's the difference in the coloration on the arms of this thing? Is it partly dead, or
Nope. It's alive. Just different color patterns, different species. Sometimes different color patterns of the same on different individuals of the same species.
Okay. And you can see on the lower right, you can see the basic. They're not roots, but
I think that's a good
the
image. I
grabbers.
think
Yep.
here.
2 feet.
Tube feet. Thank you.
Although I'm not sure those are 2 feet at the bottom.
They're
Kind of
grippers.
lasers.
They're grippers.
Lasers are on. They're good.
Well, this guy's obviously a rock climber.
So Tina Molitsova just joined us, and Tina's from Russia, from the Shirshov Institute of Oceanology in Moscow. So this truly is a worldwide team
Okay,
of
the US
scientists
group.
participating.
Yep. We're about right
We've got
underneath the
Japan,
pilot,
we've
but
got
looks like
Russia,
kind of,
we've got the U.S.,
Delta 20.
and I would imagine that we've got some folks from Guam joining us from Underwater World. It has the fantastic Exploration Command Center.
And here we're looking at some urchins. And there's one hiding down in the rock behind in the upper right-hand side. Fantastic imagery. The high-definition imagery that this ROV is able to provide for biologists. I am told by Shirley that actually, in some instances, this is good enough to identify animals right down to the species level.
Yeah. So for some for which you don't need a small piece of it to identify, to get you down
Yeah.
to species, you can use some of these morphological characteristics that we're seeing
Very
with a
top
video.
Creature crawling up.
So the yellow in he's headed toward it already.
This
He's
will be
heading it
interesting.
let's see what that is.
Let's see whether this is former
cover with that.
hydrothermal incrustation or something living
There's
in the
purple
cracks.
Stuff, purple hydrozoans or corals on the rock.
Okay, so this is another geodia, like the one we collected or videoed before. And I was just a little bit curious about and you can see lots of, looks like zoanthids maybe on the other rock. Yeah.
They're encrusting the
on
rock
that
and the sponge.
And the sponge, yeah. Very cool.
Okay, going for a medium shot.
Pilot, I
Here,
wonder
right
if
around
we
this.
might zoom in on some of the yellow material, just see if it's possible to determine if that's hydrothermal or something alive.
I think that's either coral or sponge. It looks like it might be coral.
The ship store shooting gallery is now open for the next 10 minutes. Again, the ship store shooting gallery is now open.
I see what
Keep
look like
rotating left.
polyp
Okay.
openings on the right.
But I think that was something different that's just adjacent to it.
Oh,
I can't
maybe
really
the
tell.
zoanthids?
I don't know. I don't know.
It's hard. Just up a little bit to the right, there were,
That is
Yeah, wow.
oh boy, that's
Isn't that
really
pretty?
tough
Yeah.
to tell. You think so? Yeah, to just get in that larger patch to the upper right.
Yeah, it looks like you just.
There you
Yeah.
go. I would like to be able to see the upper right tip of that rock promontory, just to the left of the sponge, because I think maybe that was where it was possible to see if there were some what looked like sponge polyp.
Yeah, so that,
Oh, yeah.
yeah, it would be coral. So, okay,
Not
so
really
now we
tall.
can see they're pulling back, trying to reposition.
And you can see another little peak in the distance, too.
We are at 363 meters.
Yeah, so you're at standstill. At
And just to
50.
give you folks at home watching to get a sense of, you know, how high up off the bottom we are, we have an altitude readout
Yeah.
that tells us we're 3.8 meters right now above the bottom.
Or above the bottom of
Yeah.
this mound. Yeah.
Right. Above the
Above-
seafloor beneath the ROV.
Or should be on the. It should be going down, decreasing in power until 1 p.m. and then it'll be nothing.
Coming in for a close-up. And you can see the pilots adjusting the light intensity to be able to maximize the best possible image. And our images are always
Yeah, they're
the
really
best
good. Really
possible.
cool.
Massive.
Yeah, it's just phenomenal. Do you think, Shirley, do you think this stuff is biological, or the yellow stuff? Well, there's tube worm.
The yellow stuff is definitely biological. I'm just trying to figure out if it's sponge or octocoral.
Okay. So would it be an encrusting kind of octocoral?
Yeah, just like those other ones that
Yeah.
we saw, those stoloniferans.
Okay.
Yeah, go left a little bit.
Ah, yeah. Look in
And now
that. Look
go
in that crevice.
up, yeah.
See? I see circular things in there. I think. What do you think? We need a flashlight right in there.
Yeah, right.
What about zooming in just
Up
the
to the-
middle? Yeah, upper left quadrant. There's a circular thing poking out at us. That has to be something alive.
Oh, there, it's alive. I'm thinking sponge. Diva, I'm going with you. I'm thinking sponge also.
I'm going to focus on the right-hand side.
Now, how could we tell for sure? I guess we could tell for sure if it sent its tentacles out, if it was...
Right. I mean,
A
if you'd be
coral.
able to see polyps, but in many cases you'll be able to see a canal structure on the surface of the sponge. Yeah.
I see what I think are lineations.
Yeah, so those are the canals. Okay.
All right.
All right. Thank you, pilot. That's great.
Wow.
Watch leads. I just wanted to check in with you. The main point of the dive, do we want to get to waypoint 2 above all, or do we just kind of want to head that way and see what we see?
We definitely want to get to waypoint 2.
Okay, got it. Thank you.
Yes, should have plenty of tether to do so. Yeah.
The topography here is pretty rough. We have a lot of these little mounds that are situated right along the crest of the ridge.
Yeah. Yeah.
So what the pilot's going to have to do in some cases is lift off into blue water and then descend a bit to be able to ride up to take a look at the next little ridge.
Yeah, we may have to hop. We may have to hop. I have a feeling that depth that the target depth was actually at the base of these big mounds, and we might be actually seeing some mounds.
To your okay, there's something to your port over there on your sonar, but and according to HiPAC, that looks to be the direction of the ridge.
Yeah.
About 45 degrees to port, actually.
Well, we are
I'd
about
say if you line up with
2 hours, right? 2 hours into our dive.
An hour and a half.
And,
Well, 2 hours from launch, yes. 2 hours from launch, exactly.
And for those of you just joining us, we're speaking to you from the Okeanos Explorer research vessel of NOAA's Ocean Exploration and Research Group. And we are out in the Western Pacific. We are diving on the flank of a volcano that is one of a string of volcanoes called the Mariana Island Arc. And these volcanoes are active. The one we're diving on is active. The area we are diving on is not active, which is a good thing. And we are proceeding up a ridge that extends to the northeast of the volcano Pagan. Pagan is known as the lighthouse of the Pacific because it erupts so frequently. It last erupted in a big eruption in 1981, and that eruption kept going until 1985, and was very similar to the eruption that took place on Mount St. Helens in Oregon.
Isn't that a slit shell at the top of that ridge? What is that? Oh my goodness, that's a slit shell.
Now, is that...
It's a gastropod.
That is
Isn't that
the
beautiful?
animals.
Isn't that beautiful? Really nice.
Wow.
Centered up anymore? Got a little bit more zoom.
So slit shells are very prized shells for shell collectors. Slit shells occur in deeper water. I've seen quite a few of them in Bahamas and Caribbean. This is the first one I've seen on this leg.
Now, wait a minute. We saw a little teeny tiny one.
You did, and I was wondering
on the top
then. I know.
of a sponge early in the dive.
And they eat sponges.
But I saw little eggs coming out of it.
Oh, then that was a hermit crab. Yep,
Yeah.
Yep. Now you actually see the foot coming out of at the base of that shell.
And there's some barnacles on it. The little white things are those barnacles.
Yep.
All
sure
right.
enough.
Hey, I'm learning.
This is beautiful.
Now, the red on the left-hand side of this thing, Shirley, is what part of the animal is that?
That's the foot.
The whole,
right over there.
Everything that we're seeing beneath the shell is a foot.
Yep.
Okay.
Yep.
So its head is going to be around the side we can't see.
Yeah, this is actually a living fossil. You know, it's a very, very ancient group of snails, of gastropods.
Wow.
And again, prized shells for shell collectors.
That is beautiful.
Siphonophore swimming towards us.
Hey, Shirley and Patty, it's Diva.
Hi, Diva.
Hey, so on leg one, we actually saw 3 foot shells, and it turns out that there are huge interest to some researchers working in the region, Patrick, Ansu, and Chong Chen. I know Chong is the champ, but I'm not sure what Patrick is. But they were extremely excited by the images, so if we could just get as much close-up images as possible. They thought that the ID was Theoretrocus teramachi, which I'll put in the event log in a second. But they just, were absolutely ecstatic and definitely said that in any case, whether it's a new species or not, or the one I just said, it was an interesting range extension if it was the species, which I just noted. So if we could just get some great imagery, we don't have to collect it. I mean, I know they would want us to, obviously, but that can be said of lots of things. But no, it's just really interesting. And this one, the color looks slightly different. So this is just great imagery, and thanks very much for taking the time to do that.
All right. Thank you, Diva.
Pilot, did you copy that? Okay, so basically you're doing exactly what we would like you to do.
Shoreside just saying, please get really, really good imagery of this. This is a rare find.
Diva Aman from the previous cruise just gave us a request for real good imagery of this, video of this, so that they can identify it for sure. They think that they know what it is, but
Tilt down so the top of the screen is at the barnacle.
and
so
look at the little barnacle waving its tentacles about.
The foot.
The scientific name of this slit shell is Barotrocus terramaki. Barotrocus. I wonder, is it named after Ted Baer? Ted Baer was a very famous, well-known expert in corals, octocorals in particular.
Okay, I think that's the best we get unless you were to move.
So I have a question.
Okay, we're good.
I have a question for you on the pronunciation of terramaki. It has 2 eyes,
Yes,
and in Latin, is it terramaki?
I think so, and that's how I would normally say terramaki.
Okay. High school
I'm
Latin is
I'm
valuable.
stretching back a long way for high
Videos
school
of
Latin.
science. If you guys can just confirm that was sufficient imaging before we move on.
Roger that. Sufficient imagery.
I agree.
Okay, roger that. Understood.
So the pilot is going to be in the water column now for a little bit, just to get to the next part of this ridge.
Yeah, we should probably get a...
And
15
as I was saying earlier, and for those of you just joining, the system that we're using for the remotely operated vehicle has another vehicle that is just above it that is then attached to the ship. So the ship will move a little bit, and the vehicle that is called Sirius, which is looking down on the ROV, then will also move in the direction of the ship. And that gives the ROV some slack for its cable, so it can move the distance between the ROV and Sirius is about 30 meters. And so the ROV can move sideways, forward about 30 meters until that tether gets taut, and then the whole system has to be moved. The ship and Sirius have to be moved in order to be able to progress forward as we continue the dive.
So there's been a nice discussion in our chat log, and Asako Matsumoto suggested that maybe the shell height of this slit shell is a little bit higher than the species that Diva was mentioning. So it could be a new species, and hopefully the imagery that we have will be sufficient to be able to identify this species, whether it's the same species that Diva just mentioned, or at least to just say that it's a new species. So, Jim, Newman is dive supervisor, and I was wondering if maybe you could explain a little bit about how we're kind of like jumping from peak to peak.
Sure. I haven't really been paying attention to topology very well, but big thing is we don't like to work down a slope. It just doesn't work well for the geometry of the vehicle.
Right. Think about when you're on in a car on a hill and you're trying to look down. You can't really see what's ahead of you.
Right. Especially if the car didn't actually tilt down with the way we don't tilt down as we're trying to go down a hill. So so in this case we've found this high point that I don't think was really expected, and so it's really a dip between two high points. And rather than try to work down to the bottom of it and then back up, we're just going to jump from one high point to the next.
Great. Thanks, Jim.
Check out the tether.
Hi, Shirley.
Yes?
This is Chris Ma at the Smithsonian. I just wanted to answer your question very quickly about Bayer Atrocus.
Yep, that would be great. Thanks.
It is indeed named after Ted Bayer.
It is good. Okay.
Oh, good to know.
So Ted Bayer worked at the University of Miami for many years, and then at the Smithsonian as a curator, and he was also a member of my master's thesis committee.
All right.
Duplicating the view on ROV HD that I see on HD2 shows a tremendous amount of that encrusting.
So DIVA is using some of the technology that we have these days to communicate in real time with others around the world, and she's tweeting a colleague in Japan to get some feedback on the slit shell.
Just give us a momentary 180.
That's an incredible thing. This is the beauty of the way this operates, is that we on board the ship can provide some input with regard to what we're looking at, but if it's not our field, we have a backup on shore with scientists who are experts in a whole variety of fields, and they're contributing not only while we're diving, but these are the people who have written the proposals, the plans for the dives that were submitted to NOAA, and they then are discussed in a group chat that can include 140 people.
Yeah.
And we are looking for consensus with regard to what aspects of a particular area might be of interest to people. More encrusting stuff.
So
What's
are
the
those all
stuff?
I'm not sure if they're all hydroids on that rock.
Oh yeah, sorry.
Okay, look at it now. Shirley. There. Oops.
Yeah.
Just past it
Yeah,
by.
that's
There's some
down.
fingers, thin fingers sticking up that are also encrusted with that. Like about the thickness of a pencil or so.
Yeah, boy, if I didn't know better, I would say that there's an encrusting sponge under there, but I don't think that's...
But that's
I don't
just
think-
you.
Yeah, I know. I don't think those projections are probably hydroids. They are weird, aren't they?
Yeah, I have no idea what that is.
Here on the left.
Okay, they look like teeny tiny cotton swabs, and out of the top of them are little hairs sticking
Yeah,
up.
I'm thinking hydroids.
Now, do you see the ones that look like they're
Yeah,
encrusting a
over to the-
stalk of
yeah,
something?
Yeah, yeah.
Okay.
Any of those.
Hydroids. What is a hydroid?
Well, it's in the same phylum as corals.
Okay.
and Shell. It's a hydrozoan, so it's in that same phylum, the phylum Cnidarium.
Huh, okay. So they're grabbing
Space.
grabbing
Yeah, space.
they certainly
They're looking for real estate.
they have all of the real estate in the field of view at the moment. So they're feeding on things that are floating through in the water, little
Yes.
plankton and
Yep,
stuff.
yes.
Okay.
And if you ever run across these in shallow water when you're scuba diving, they sting like heck, let me tell you.
Do they?
Oh, yeah. Really.
Wow.
And sometimes you can't really tell that that's what's in the sample that you're touching.
Huh. Well,
wow.
When we get the rock back, we're going to make sure to wear gloves. Wow, they are everywhere.
Mm-hmm. Very different from what we saw yesterday. Of course, we're shallower than we were yesterday as well.
That's true. So we're getting a good feel for the.
Pilot, can we go and look at those yellow branches ahead, please?
We're getting a good feel for the distribution of organisms
and how patchy
as a
that
transect
can be.
in depth. Zooming in, and that's more hydrozoans.
Oh, so it's the same. Okay, so I'm trying to figure out what that.
It's a plague.
I don't know.
A plague of hydrozoans. Even the branching thing on the left-hand side.
I'm actually thinking that yellow is a yellow encrusting sponge on the rock, and then the hydroids on top of it. Okay, that's good video.
So are all these projections just things that the hydrozoans are colonizing?
Yes. Mm-hmm.
Okay, so there aren't any corals. I don't know though, Shirley. The yellow corals on the left-hand side of the screen now, are those
could
corals?
be? Yes. Yeah. Let's.
Maybe we should take a look.
Yeah, pan over to the left. I think it might be the same that we saw.
Or the yellow one that's right in the center bottom view.
Yeah, whatever is easier for the pilot to get to.
Right.
You ready? Okay.
Oh, there we go. There's a coral. Right next to a dead
beautiful.
coral. And what is that orangey thing? Is that another one of those crabs?
Squat. It's a
squat lobster? That's a squat lobster.
And probably a Nalipsamia, so same coral that we saw earlier.
Okay.
Video whenever you're happy with that image.
We'll need to get several seconds of this.
That's fine. Yep, that's fine. So part of what we try to do is get several seconds of video imagery because we're using the video to take screen grabs to have some still images as well. So it gives our videographers an opportunity to select the best screen image out of that screen grab out of their video.
And all of that video that we are collecting is available to the general public. Everything that is done out here on the Okeanos Explorer is for the community at large. So not only scientists, but anyone in the public who might like to take a look at any of these images, they are freely available. No copyright. And you can use them on Facebook or Twitter or whatever you like.
So those, I'm not sure, but they may be just young sponges, but I'm not sure. Those little pinkish white. Very nice.
Wow. Thank you, pilot. Thank you, videographer.
Okay, we can go up. Okay, let's
mosey on.
Yeah, go ahead up. That's good.
The rocks on the seafloor that you're looking at that are making up these mounds are very likely what we call pillow lavas.
Left there.
And pillow lavas form as magma is erupted at the surface of the seafloor, contacts the cold seawater around it, and basically freezes. So very often if you collect a rock from an area like this, it will have a glassy rind that can be a centimeter thick or more. The rock that you see in the left-hand side of the screen, right next to the white sponge, has the typical sort of keystone form. These lavas form tubes that look like toothpaste squeezed out onto the seafloor, and as they cool, you get cooling cracks that are radially distributed within the tube itself. So very often they are kind of pie-shaped when you see them on the seafloor. And we are zooming in on a sponge and a barnacle.
Yes.
Hey! Cool. The geologist knows a couple of terms.
Yeah, more than a couple.
Is that a live barnacle?
Oh, it looks like it
looks dead
to me.
Yeah. The sponge doesn't look too happy either. But now this guy's different from the one we saw yesterday.
Well, the one we
other
saw
one
yesterday
had a stalk.
Exactly. So they're different kinds.
Are those different species? Or
Absolutely.
Okay, so this guy will never grow a stalk.
No.
Ha! Stuck to the rock forever.
So are the ones with stalks.
Our pilots are switching out here so that they get a little bit of a break. Fresh eyes. Fresh arms. But they're always very polite.
I think we can proceed along our dive track.
Yep, video's clear.
Yeah, it's amazing how bumpy this ridge is that we've chosen on the bathymetry. The topography of the seafloor, the map that is created that we use to decide where to pick a dive site, showed a fairly smooth slope on this ridge as we go up. But you know, once you get down on the seafloor, that's when you get the reality check. And the reality check here is it's a bumpy road.
So yeah, the current's been due west lately, so if you're facing our direction, you have to lateral a little while you go forward, or you can it'll naturally spin you with the tether drag so that you're facing due west. So
when the road is bumpy like this, sometimes the pilots have to just kind of fly from one of these little mounds to the other, because they cannot see behind themselves. So imagine yourself backing down a real steep driveway, and your rear view mirror is broken, and that makes it tough.
But fortunately, they have Sirius.
They have Sirius. That's right.
We've got Sirius.
The vehicle that is tethered immediately above the remotely operated vehicle gives the pilots a larger field of view.
The last one that was set was
but even so, if they were trying to go down a slope with the back end of the vehicle pointed toward that slope, could be dangerous.
So we are at 368 meters, and we're proceeding along our agreed upon course, and we'll keep doing surveying across the bottom. Our target end depth is about 250 meters, so we still have plenty of time. This is an 8-hour dive, deck to deck, from launch to recovery. So, and we're in fairly shallow water, relatively speaking, and so we'll have quite a few more hours to work along this ridge.
If you take a look at the Sirius view, I think that's the second option
Yeah,
on the bottom
right
of
side.
your screen. If you're looking at the video at home, you'll notice that it's really dark on either side of the ROV, and that is because, as you can see on the main screen, there are really steep drop-offs
Something
along
looks out
the
of
edges
place.
of this ridge.
Yeah, that is something that should image.
Now, if you want, you can probably move me up there. He's getting. Yeah, what do
The
you think?
pilots have the
20 meters?
laser beams
30
on
meters?
right now on the seafloor, and that's for
I'd say,
scale.
well,
So the
can we start
distance
with 20
between the
just for now?
two red
Sure.
dots that
At
are
your
on
heading?
either side
yeah,
of a
I'm at
little
215.
white branching,
So if
oops,
we
now
can
it's
move forward
off
and
to
step
the
down
right
image
of a
that.
little branching, probably a coral. Those are 4 inches, 10 centimeters apart. It's about 4 inches.
Bridge, this is nav. We would like a ship move 20 meters, bearing 215 degrees at 0.2 knots.
So there's a dark spherical critter just
Good
at
copy,
about
Bridge.
1 o'clock from that branching white thing. And Shirley, do you think that's a sponge? Or is that a holothurian?
What branching white thing?
See
Oh,
the
down to
branching
right,
white?
on the right-hand side?
Not on the lower right, but kind of mid-screen, slight
Rich.
to the right of center, and then to the
Yeah,
upper
I don't know.
right of
Might
that
be those
animal.
dead branches again.
No, it's
Oh, wow.
it's not a holothurian.
Wait, what the heck?
that?
Wait, what the heck is
What
that?
is that? We got something I've never
Wait,
seen
no,
before.
What is that?
What is that?
A very
Okay,
full
people on
anemone.
shore, help!
No pressure, Sean.
What is that thing?
I'll get a little
They,
closer.
they seem excited. You could just hop there now.
It's got
It'll be
polyps.
Yeah, Tina, what do you think? She asked us to kind of zoom in on this as well.
Yeah, I think the pilots are getting the ROV into a
Yep.
position where they can be stable, because there's a pretty, well, there was a pretty strong current. It dropped out for a bit. I don't know if it's still really strong, but it was making things difficult early in the dive for the pilots to
Get a little bit
orient
further left
the
right
vehicle
now.
so that we can get a close-up
Plus
view.
we're moving.
This thing has a red tone to
brown,
it.
yeah, reddish brown.
And little white polyps. And there's a sea urchin over to the right on a beautiful cross-section of a very large pillow lava fragment.
So Tina is thinking, no, new to Brank, and then she says no.
Lasers off.
Yeah,
Wow.
stumped the people on
Wow,
shore as
I
well
don't
as
know.
us.
Wow, this is very cool.
This is a gorgeous
Break your
thing.
camera, let me know.
Oh, look. Is it an anemone?
Man, I
It's
don't know.
not, oh my gosh.
How wonderful. Just look at this thing. Holy smokes. This is beautiful.
So Diva Amon thinks it's a young pom-pom anemone.
Wow.
Yeah.
Liponema, she says, question mark. And Tina says, looks like.
Wow.
This is a beautiful,
Oh, isn't that really nice?
beautiful thing.
That's really nice.
Want to pull out for a full species?
We're being quiet here as we scribble down notes
I am.
really fast. Oh my goodness. Something new. Has anybody seen one of these things anyplace else? Obviously, Tina.
And Diva, probably.
Yeah,
'cause,
Diva
wait, I'm
identified
gonna look at
it.
Let me look at my
but
little
where,
cheat sheet here.
Diva, are you online? Can you tell us where this thing was seen before?
What did I do with my... She says no.
She says no.
She's
Never,
never like this.
Wow.
Let's look down at the shadow. Seeing if we can see any kind of feet.
A pom-pom anemone. I like that.
If we can.
Certainly looks like a pom-pom.
It does.
Were you a cheerleader in high school?
I'm not going to tell.
80 degrees now with camera. How far?
I think you would have
Looks
been
like the
really
ship
good.
I was the
stopped.
captain of the cheerleaders.
So you're
All
gonna go a
right,
little further.
all right.
Since you're on top of them, I would come up to like 20 meters.
I
depth or-
certainly
That's
can
a long
believe
time.
that.
That's a
Where you know
long
where the
time
tether
ago.
is.
I think it
Yes.
shows because you guys have me very excited about this. You guys are definitely a good cheering.
Well,
That's
I
not my single
confess.
color, though. Were you?
I was on the squad. There are a couple of ding dongs out here, you know.
No, but that just shows that we're
You
not
know-
like total nerds.
Well, no, it also shows that the scientist must have enthusiasm. So any of you girls out there listening, you know, just keep up the enthusiasm.
This is lovely. Really lovely.
Well, that's what I
See
tell.
those, those lasers. So you can see the disc, how big this is. Those lasers are
10 centimeters.
10 centimeters apart.
So this guy is about 7 inches in diameter.
In diameter,
So.
yeah. Really cool. Very nice. Well, wait, stand by. Stand by. Do we want to collect this?
Oh my goodness, there is a question. Okay, Diva, Tina, what do you think about collection?
Do we need to collect this, or do you think we can identify this from the excellent imagery that we have? Stand by. There's just a little bit of a delay.
Well,
And
at least it's obvious how you're going to grab it, because it's a perfect sphere. You don't have many options.
Is it still difficult for people in the ECCs to hear us? I should probably write that down. Is it?
Yeah, it's fairly straightforward as long as
Please don't kill
if
it.
you do it in grip force 0 or 1, and then you just, instead of when you gotta let go, just like drop it. Don't try to put the jaws in and then feather it. Just like
What drawer would you want to try to get it into?
the easiest one, yeah.
While we're waiting for that, do you want to just tilt up and we'll look at that yellow thing at the top?
And we have I have no idea how hard it is to be.
yeah, it might be kind of squishy, but I don't know.
Diva is voting
Okay,
to collect. She
okay.
says she thinks it's new.
Okay, well, I'd be willing to bet that just about everything we're seeing here, even the ones for which we know the genus names, are probably new too.
So,
Pilot, request that you collect this, please.
You think it's going to be possible to extend the bio boxes here on this
Yeah, they'll
slope?
do-
Is that
yeah,
going to be
they'll-
hard for you?
Good.
that looks good.
Thank you so much.
Co-pilot, you comfortable with your movement? I think you stopped a minute
You mean
ago.
this one?
Yes, I've stopped.
Okay.
Thanks.
For collections, the HPU gets ramped up all the way? Or
You know
just
what,
three-quarters?
I'm not bringing out my best
A little
moves
more than
yet.
3/4.
Okay, roughly where I
Hey,
have
we
it.
got to get them coordinated, lady.
Now that we know.
The thing that I tell, I bet you
can
do
set
this
up the
too,
pins
Shirley. You know,
for
when you go to talk
Okay.
to young
Especially four
kids,
drawers and
sometimes they're
You
kind of
retract both
intimidated by,
full
you know,
drawer.
oh, here's
Extend
a
the one you want
scientist. Oh
open,
dear,
and then
you know.
you're ready.
And
Okay.
when I tell them, ask them generally, what do you think is required in order to be a scientist? And I get,
looks like your drawer
you got to
can
be
open
really
for sure.
smart.
You got both skids down,
you got to
so you're
have
pretty stable.
fuzzy white hair. They have these impressions of
looks stable,
what is really needed. The basic thing that a person has to have in order to become a scientist. And really, the basic thing is unending curiosity, enthusiasm. Stick-to-itiveness is also required to get through grad school. But
lost, you're not-
but if you've got
So it's nice.
the enthusiasm to learn things that is going to last you your lifetime, then
I'm gonna
not
make
only
sure that
can you be a
everything's
scientist,
right
but
on here.
basically you are a scientist.
I'll slow at this part. Okay, now I can extend.
So it's no wonder
we're
the two of us were
putting it in the port
cheerleaders.
vial. Extending port vial pen.
And you did a drawer in before, right?
I did. I lifted
Okay.
the pins,
Yeah.
and I brought the drawer in, and now